Huntington disease, an autosomal dominant genetic disorder, is a progressive brain disorder that causes uncontrolled movements, emotional problems, and a loss of thinking ability. Adult onset is the most common form of Huntington's disease. Early signs and symptoms include depression, small involuntary movements, irritability, poor coordination, and trouble learning new information or decision-making. A number of people with Huntington's disease develop involuntary jerking or twitching movements known as chorea. Chorea is caused by an excess of the neurotransmitter dopamine, and tetrabenazine reduces the activity of dopamine in the brain. As it progresses, these movements will become more pronounced. The affected individuals may begin having trouble walking, talking, and swallowing. They will also experience changes in personality and a decline in their thinking and reasoning ability. Most people with Huntington's disease develop signs and symptoms in their 30s or 40s, but the disease may emerge earlier or later in life. Individuals with Huntington's disease will often live about 15 to 20 years after these signs and symptoms begin. HTT is the gene that is responsible for producing the protein called huntingtin, located on chromosome 4. The function of the huntingtin protein is not known for sure, but it does play an important role in nerve cells. Within the cell, huntingtin may be involved in signaling, transporting materials, binding proteins and other structures, and protecting against apoptosis. The protein is required for normal development before birth and is expressed in many tissues in the body, with the most being expressed in the brain. The mutation of the gene HTT is what causes Huntington disease involves a DNA segment known as a CAG nucleotide repeat. CAG is made of cytosine, adenine, and guanine nucleotides that appear numerous times in a row. Normally, CAG is repeated 10 to 35 times, whereas with people with Huntington disease, the CAG repeat is repeated 36 to 120 times. However, if the CAG is repeated between 36 to 39 times, the signs and symptoms may not develop. In order for Huntington's disease to occur, the abnormal repetition of CAG has to occur, meaning that the gene HTT is therefore mutated. Once this repetition occurs, the protein huntingtin is then made in excess with an abnormally long polyglutamine tract at the N-terminus, causing the protein to be unstable. Enzymes within the cell oftentimes will cut the elongated protein into fragments. These protein fragments then form abnormal clumps known as neuronal internuclear inclusions, NIIs, inside nerve cells and may attract other normal proteins into the clumps. NIIs are considered as a coping mechanism opposed to a pathogenic mechanism to stem neuronal death by decreasing the amount of diffused huntingtin. This process is likely to occur in the frontal cortex, the part of the brain that controls thinking and emotions. The loss of neurons mostly occurs in the basal ganglia. The second most part affected is the cerebellum cortex, which controls movement, perception, memory, and emotion, which is why the symptoms of Huntington's disease are the way they are. As HD progresses, it will spread throughout the brain and affect the control of hormones. There has not been a case of Huntington's that has occurred with less than 36 repetitions of CAG. However, depending on how many repeats you have determines the severity of the disease. If there are more than 60 repeats, then that person develops a more severe case of HD, known as juvenile Huntington's disease. And if there are less than 40 repeats, that person is likely not to experience any of the signs or symptoms. As the mutated gene is passed down from generation to generation, the size of the CAG repeat expansion can change. It will often increase in size if it is inherited from the father's side. There seems to be a lot of research going on for Huntington's disease. There are numerous medical trials for the disease to try and find a cure. The cause of Huntington's has been known since 1993, but it is difficult to prevent a disease if you do not know what the cause of it looks like. It has been known that the protein huntingtin is the cause of the disease and what the protein does to the brain, but up until recently, nobody knew what exactly the protein looked like. Now that they know what it looks like, they can begin looking for ways to treat it and prevent Huntington's disease from occurring. While there is a lot of research for Huntington's disease and what causes it, in the long run it is an incurable neurodegenerative disorder caused by the dominant mutation of HTT. All of this research goes to better understanding the disease and why it happens, along with trying to find a way to prevent it from occurring. Even though they are not able to cure Huntington's, they are able to alleviate the symptoms caused from it and even prevent it from occurring in future generations. There isn't a treatment to modify the neurodegeneration in humans, but tetrabenazine can reduce some movement abnormalities. Some current trends in this research are a focus on the CAG repetition over exon 1 and the pathogenesis of the HTT gene that makes this disease so devastating. There is no cure for the disease yet, so much of their research is devoted to finding out how to reverse the mutation of the HTT protein or prevent the incorrect splicing of HTT exon 1. This research is important because this is an inherited neurodegenerative disorder that currently has no cure, and if a cure for this disorder can be discovered, perhaps it can be linked to the cure for multiple other inherited neurodegenerative disorders, such as neurofibromatosis 1 and 2, frontotemporal dementia, and von Hippel-Lindau disease. A journal article published in 2017 titled "The Pathogenic Exon 1 HTT Protein is Produced by Incomplete Splicing in Huntington's Disease Patients" discusses how exon 1 of the Huntington gene does not always splice to exon 2, which results in the production of a polyadenylated mRNA that encodes the exon 1 HTT protein responsible for this disease. The authors of this article have developed a specific protocol that allows for the quantitative analysis of the HTT exon 1 transcription levels to be compared to a series of fibroblast lines and brain samples that reveal that the HTT exon 1 mRNA is present in fibroblasts and the motor cortex, hippocampus, and the cerebellum of people with Huntington's disease. The methodology of this research started with RNA extraction from patient-derived fibroblasts, and the use of post-mortem human brain tissue samples was approved. Then, the RNA samples collected were then reverse transcribed and repeat-sized. Mice were bred and genotyped, and a DNA sample was collected via ear punch from which RNA was extracted from. Immunoprecipitation, Western blotting, and immunoprobing for the mouse tissue was performed, and all the data was analyzed and compared. The main findings of this research were that HTT exon 1 is produced in the brains of those with Huntington's disease, specifically those with early onset, and that the severity of the disease may be directly tied to the missplicing of HTT. Some implications of this research were that there could be new strategies that can be used to lower mutant HTT levels in patients using this research. All in all, it is clear that the mutation of the protein huntingtin is responsible for Huntington disease. The severity of the disease all depends on the amount of times CAG is repeated. The cause of this repetition is still unknown, and there is still no cure to reverse the effects of Huntington's disease. Thank you for watching.